Molecular Genetics. Part 2. UGC NET Anthropology. Welcome to AnthroNet Academy, a study aligned with the UGC NET Anthropology syllabus. Description box. Please visit the description box for books references. Dear learners, if you find this content helpful, please consider subscribing to AnthroNet Academy. Your support really means a lot and keeps me motivated to create more valuable content for you. Big surprise at 500 subs. Once AnthroNet Academy hits 500 subscribers, we're dropping something truly special just for you. A full video revealing 100 powerful, underrated, and must-read books list in anthropology, complete with author names and insights important for UGC NET exam. Test yourself. Don't miss the final segment of this video. It includes high-yield MCQs that could make or break your next anthropology exam. 1. RFLP Restriction Fragment Length Polymorphism. What is it? RFLP is a technique used to detect differences in DNA sequences by cutting DNA into fragments using special enzymes called restriction enzymes. How does it work? A restriction enzyme scans the DNA and cuts it at specific sequences, e.g., G8TC. Everyone's DNA is slightly different. Some may have more or fewer of these cut sites. This produces fragments of different lengths for different people. When run on a gel electrophoresis, you see a pattern of bands. These patterns are unique to each person, just like a fingerprint. Use forensic science for DNA fingerprinting, paternity testing, genetic disease detection. 2. VNTR, variable number of tandem repeats. What is it? VNTRs are short sequences of DNA, 10 to 100 base pairs, that repeat multiple times in a row at specific locations in the genome. Example: if the sequence GATA repeats, one person might have GATA, 3 repeats. Another person might have GATA, 5 repeats. Why is it important? The number of repeats varies greatly between individuals. This variation is inherited from parents, so it's used in identification. Used in early DNA fingerprinting techniques. Important in forensic analysis and genealogy. 3. STR, short tandem repeats. What is it? STRs are like VNTRs, but the repeating units are shorter, usually 2 to 6 base pairs long. Why STRs are preferred over VNTRs? STRs are more abundant in the genome. Easier and faster to analyze using modern machines like PCR, polymerase chain reaction. Example: if the sequence at repeats, one person might have ATATAT 3 repeats. Another person might have ATATAT 6 repeats. Use. Most modern forensic DNA profiling systems use STRs. Used by FBI's CODIS, Combined DNA Index System. 4. SNP, Single Nucleotide Polymorphism. What is it? A SNP is a variation in a single nucleotide, A, T, C, or G, at a specific position in the DNA. Example: at a certain position, one person may have A, another may have G. This is called a SNP. How common is it? Occurs every 100 to 300 base pairs on average. Over 4 million SNPs are known in the human genome. Use. Understanding disease susceptibility. Personalized medicine, how people respond to drugs. Tracing ancestry and evolution. 5. Mitochondrial DNA, mtDNA. What is it? Mitochondria are the energy-producing organelles in cells, and they have their own small circular DNA separate from the nuclear DNA. Key facts. Inherited only from the mother. Each cell contains hundreds to thousands of mitochondria. Mitochondrial DNA has 37 genes, and it's very stable. Why is it important? Used in maternal lineage studies. Useful in ancient DNA analysis, bones, teeth. Helps solve forensic cases when nuclear DNA is degraded. 6. Genic mutation. What is it? A genic mutation is a change in the DNA sequence of a single gene. Types of genic mutations. Point mutation, change in a single nucleotide. Insertion, adding extra nucleotides. Deletion, losing a few nucleotides. Missense mutation, changes one amino acid in a protein. Nonsense mutation, introduces a premature stop signal. Effect, can lead to non-functional or harmful proteins. Examples: sickle cell anemia, cystic fibrosis, hemophilia. 7. Genomic mutation. What is it? A genomic mutation involves changes in the entire chromosome or sets of chromosomes. It affects the whole genome structure, not just a single gene. Types. Aneuploidy, gain or loss of a chromosome, e.g., Down syndrome equals 3 copies of chromosome 21. Polyploidy, more than 2 complete sets of chromosomes, common in plants. Chromosomal rearrangements, deletion, duplication, inversion, translocation. Effect, can lead to developmental disorders, infertility, or even cancer. Summary table. Term full form, what it measures, use inheritance/impact. RFLP restriction, fragment length, polymorphism, differences in DNA cut patterns, forensics, disease unique patterns in individuals. VNTR, variable number, tandem repeat number of longer repeats, forensics, paternity, high variability. STR, short tandem repeat short sequence, repeats, modern DNA fingerprinting, more precise, widely used. SNP, single nucleotide polymorphism, one-letter DNA variation, ancestry, disease risk, very common. mtDNA, mitochondrial DNA, maternally inherited DNA lineage tracing, forensics from mother only. Genic mutation, mutation in a single gene, genetic diseases, alters proteins. Genomic mutation, entire chromosome changes, syndromes, cancer, large-scale impact. Test your knowledge. Q1. Which of the following genetic markers is most suitable for forensic identification due to its high variability and ease of analysis using PCR? A. VNTR. B. SNP. C. STR. D. RFLP. Correct answer, C. STR. Explanation. STRs, short tandem repeats, are highly variable between individuals and are short enough to be efficiently amplified by PCR. Hence, they are widely used in forensic DNA profiling. Q2. What is the major difference between RFLP and STR analysis? A. RFLP uses mitochondrial DNA while STR uses nuclear DNA. B. STR uses restriction enzymes while RFLP does not. C. RFLP analyzes fragment length after enzymatic digestion, while STR uses PCR to analyze repeat length. D. STR is based on SNP variations. RFLP is not. Correct answer. C. RFLP analyzes fragment length after enzymatic digestion, while STR uses PCR to analyze repeat length. Explanation. RFLP involves cutting DNA with restriction enzymes and analyzing fragment length. STR uses PCR to amplify short repeat regions and determine the number of repeats. Q3. Which of the following is maternally inherited and is useful in tracing maternal ancestry? A. Y chromosomal DNA. B. Nuclear DNA. C. Mitochondrial DNA. D. SNP. Correct answer. C. Mitochondrial DNA. Explanation. Mitochondrial DNA is inherited solely from the mother and is useful in maternal lineage and ancestry studies, especially in ancient DNA analysis. Q4. A single nucleotide change in a DNA sequence that may or may not affect protein function is known as A. STR. B. VNTR. C. SNP. D. Genomic mutation. Correct answer. C. SNP. Explanation. A. SNP, single nucleotide polymorphism, is a change at a single nucleotide position. It may be silent or may cause changes in protein function depending on its location. Q5. Which of the following best describes a genomic mutation? A. Point mutation in a gene. B. Frameshift mutation in an exon. C. Duplication of a chromosome segment. D. Insertion of a single nucleotide. Correct answer. C. Duplication of a chromosome segment. Explanation. Genomic mutations refer to large-scale changes involving whole chromosomes or chromosome segments, such as duplication, deletion, or aneuploidy. The other options are gene level, genic, mutations. Thank you for watching. Please like, share, and subscribe to my YouTube channel, AnthroNet Academy, for more insightful content on anthropology. Stay safe and connected.